So it's my pleasure to introduce you to NAVAN Technologies. My name is Michael Nowak. I'm President and CEO of NAVAN Tech. In the next few minutes, I'd like to describe to you a novel technology platform that we've exclusively licensed from Stanford University. We originally were funded using NSF through a generous grant from the NSF, an STTR grant. Today we are headquartered at J Labs at Oyster Point in South San Francisco, and it's my pleasure to announce that today that we've closed seed financing led by Amgen Ventures, by HTGF, leading European venture capital firm, Hemi Ventures from Palo Alto, California, and other institutional and private investors. Our technology is focused on addressing what we believe is a key bottleneck in developing the next generation of cell and gene therapies. It's the manufacturing of transformed cells. In the next few minutes, I'll describe some of the progress we've made together with some strategic collaborations that are in place. So I'm not going to belabor these issues so I can mainly describe to you the technology, but we're addressing key manufacturing issues that we believe are critical to the development of this next wave of innovation. And that's not just delivering interesting cargo into hard-to-transfect cells, and by that I mean everything from oligos and DNA and RNA to complete transposon systems into human primary cells, but doing it in a way that maintains cell viability and has the cell functionality that one is looking for, and doing it in a scalable fashion where you don't run into the cost or technical or safety issues that you do with current technologies. In this picture here in the middle, you can see these beautiful, what we call nano straws that basically are like microscopic, well actually nanoscopic syringes that we grow on top of pores and semi-porous membranes. And so in the diagram on the left, you can see there's a fluidic channel where one can put in whatever cargo one wants. The beautiful thing about a physical delivery system is we don't really care which cell you're talking about or which cargo you're trying to deliver. On the other side of this plastic membrane and on top of the straws, one can put whatever cell one wants, and through an intimate contact between the cell and cell membrane and those straws, we're able to inject any cargo into any cell with full dosage control, but to do it in a very non-perturbative fashion. And I think if you saw the diagram before, and we'll see in future diagrams, that the dimensionality is such that this is really the equivalent of a needle in the arm of you. In this example, we're a needle in the cell. So how do we get to viable and sustainable cost of goods in next-generation cell and gene therapies? We believe one has to tackle the delivery challenge. One has to do it with a viable, in the end, producing a viable cell and in a scalable, cost-effective manner. Just to demonstrate quickly some of the cells and cargoes we've been able to deliver, everything from T cells, and I don't have B cell data there, but to macrophages and other monocytes, and also to stem cells, and we've been able to deliver with a variety of efficiencies. We haven't really optimized yet. Part of the plan for this year is to optimize some of the efficiencies and viability numbers. But you see, in all these transformations that we've been able to successfully conclude, we have very high cell viability. And we believe that's a topic that one is just starting to hear about, because cell quality is hard to measure. It's controversial on how one determines what a fully functional and unperturbed cell is. But the quality of your cell therapy in the end is going to be dependent on the quality of the cells that result from your manufacturing process, and we believe that's our core strength and capability. I won't go through the physics of how our technology works. We do use a small electric field to enable the delivery of the cargo into a cell. You can see the sort of intimate contact is diagrammed on the left there. Sometimes, depending on the dimensionality of the straws, the straws penetrate the membrane. Sometimes we electroporate a small hole in the membrane, but directly above where that straw leads. So you truly have an injection of material into the cell. We typically use tens of volts across this, so it's not an electroporation technology where you're using hundreds of volts and destroying lots of cells. And we know that because we can crank up the voltage until we do kill the cells. What's important to note, though, is we have full control of the dimensionality and consequently full customizability capable for us. We can control the width of the straws, the length of the straws, and the density of those straws. So we believe that'll lead to optimization capabilities. I won't go through this in much detail, except we believe that by our technology, using our technology, one's able to address the high cell mortality rates or the cargo limitations that are currently involved in a variety of different competitive technologies, as well as some of the safety and cost issues. And that competitive advantage manifests itself in a variety of different ways, whether that's controllable dosage delivery. We've talked about the versatility of any cargo into any cells. It's important to note our typical transfection time is 40 seconds, so we're able to do this very quickly. By having the high cell viability, we're able to do multiple deliveries, and that's multiple deliveries of the same cargo or multiple deliveries of delivering different cargo at different times. And I have no time to talk about it today, but we're actually also able to sample from these cells, and there's possible assay tool products in our future where we're able to assay living cells and so consequently avoid destroying cells in a quality control or quality assurance module, but also to do some very interesting science. And one of the other ways we know we're not perturbing or damaging the cells is we're actually able to sample cells we've transfected and demonstrate through RNA and genetic analysis that there's none of the stress indicators have been lit up. So just a little bit more data. We've been able to demonstrate similar delivery efficiencies in all PBMC subsets. What's interesting on the right-hand side there is that's data from unstimulated CD4 positive cells, and after transfection, after delivery, those cells are still unstimulated, which we believe can offer some interesting therapeutic potential. One of the big questions, of course, is can you do this in a durable, can you enable a durable gene editing? And indeed, we have a couple examples. This is with collaborators from UCSF. We've been able to do a RAB4 gene knock-in. In this case, it's Jurkat cells using Cas9 RNPs or mRNAs. And 11% efficiency, that's actually a factor of 5 better than what people are doing with traditional electroporation technology. So in an unoptimized experiment, we were very, very excited to see that sort of result right from the start. Conversely, or with a different sort of cell, we're in this case using hematopoietic stem cells, we were able to do use Cas9 mRNA to do a CD45 knockout, again with similar, in that case, 10% of efficiency of the knockout. Well, you can see we've had very high delivery of the Cas9 mRNA, and throughout all of these, very high cell viability. So I don't see a clock here. I don't know how much time I have left, but 5 minutes? Thank you. So very quickly, I want to just go through some of our commercialization strategy. I think the FDA has correctly identified manufacturing, unlike in current biotherapies where clinical development has been the primary challenge. In this next wave of innovation, Scott Gottlieb pointed out that he believes 80% of the risk, 80% of the review time is going to be spent on manufacturing and quality issues. And that's what we're directly addressing. We've got a number of competitive advantages I don't have time to go into. Would like to point out that since our technology is based on semiconductor technology using decades-old technology, that leads to low-cost supplies, low-cost equipment, and vast scalability capabilities. I've mentioned how we have got controllable geometry allowing customization, and we're cargo agnostic. We're able to deliver interesting molecules on the left into interesting cells for a variety of important cell outcomes, whether it's gene editing to pathway modulation, and consequently enable a number of cell-based therapies or regenerative medicine therapies. I'm not going to spend any time on this, except we do believe that the market opportunity is vast. What we're focused on is to enable the industry to move away from the centralized facility approach to cell and gene therapy to a modular point-of-care solution. Our footprint is actually very small for our gene delivery technology, and provide a module initially, and then ultimately an fully integrated closed system to be able to do that. And consequently, our commercial strategy is to develop and deliver not only those modules and systems, but also to go deep into the immuno-oncology and regenerative medicine space and really have indication-specific efforts and to participate those via partnerships, joint ventures, and through our own proprietary programs, leading to a pipeline over the next few years that looks like these. So these are current efforts in our corporation. We've got a number of T-cell efforts with a variety of different commercial partners. These are collaborations right now, not partnerships, but whether it's T-cells, B-cells, or macrophages, we've got ongoing delivery efforts, as well as on the regenerative medicine side with stem cells and fibroblasts. And so we're looking forward to a 2019 that has a number of significant development milestones. As I mentioned before, optimizing cargo delivery on the transient expression side, but also on the durable side with Cas9 or Sleeping Beauty transposon systems. And but importantly as well, to scale up production capabilities, both in terms of nanostraws and both in Europe and the US, but also in terms of consumables and in the research device we have today, and developing a clinical scale product for use with our strategic collaborators and partners. Couldn't do this without a phenomenal team of scientists and business leaders, as well as a deep bench of advisors and strategic collaborators. So thank you very much for your time. Happy to take any questions, but we believe the key to the next generation durable, effective cell and gene therapies is producing quality cells, quality therapies, and that's by delivering them without damaging the cells. Thank you.